In part 5 of this series on microbial growth, we talked about how labs make calculations to predict the number of bacteria that result after one or more divisions by binary fission. We also looked at some equations to calculate generation time. Finally, we looked at growth curves that scientists use to follow the growth of bacteria over time. The question is, where do those numbers come from? For example, in one of the practice problems, you were told that a culture had 1,720,320 cells after incubation. But where did that number come from? We'll tackle that question plus a few more in this, the final video on microbial growth. Your sole objective for this video is to describe 4 methods for measuring bacterial growth in a lab. As you can see from this slide, there's a number of methods that can be used to measure microbial growth, and not all methods are even represented here on the slide. Generally, we can divide the methods up into 2 main categories. You have the direct methods that measure individual cells or the colonies that result from cells. Then you have the indirect methods that don't measure cells or colonies, but instead they measure some characteristic indicative of growth. And based upon that characteristic, we extrapolate how many cells there have to be. In this video, we're going to look at 2 or 3 direct methods, and we'll look at what I consider to be the most common indirect method. When we have bacterial cultures in the laboratory, there can be millions of cells within our test tubes. To calculate how many cells there actually are, plate counts can be useful. In a plate count, we're going to take a sample of the bacteria from our tube, we're going to spread it out across an agar plate and grow colonies. Now remember, each colony represents one cell or a cluster of connected cells. So, by counting the colonies, we can get a good estimate of how many bacteria there were in our test tube. But remember I said there were millions of cells? So that means that our specimen can have hundreds of thousands of cells, and those are too many to count on a plate. So the first thing that we need to do is a serial dilution. We're going to systematically reduce the number of cells in our specimen that we're going to put on the agar plate. And by doing this systematically, we're making it so that we can calculate how many cells there originally were. I know that sounds confusing. Let me explain it first. Here is a serial dilution experiment. We're going to start out with 5 test tubes that all have 9 milliliters of culture medium in them. Okay. So here's our first test tube. And to that test tube, we're going to add 1 milliliter of inoculum from our culture. When we add that 1 milliliter of inoculum to the 9 milliliters of sterile medium, we get a total of 10 milliliters. Then what we would do is we would mix the cells from our inoculum up with the medium. This would give us a 1:10 dilution. 1 part of this comes from our inoculum, and our total volume is 10 mils. After we've kind of mixed the bacteria up with the culture medium well, we would take a 1 ml specimen or aliquot from this tube and we'd add it to our next tube. So remember there's 9 milliliters of sterile broth here, so when we add 1 milliliter from the other test tube, we now have a total of 10 mils. So we would mix all of that up. So this is basically a 1:10 dilution of a 1:10 dilution. You're probably wondering, how is this a 1:10 dilution of a 1:10 dilution? Well, remember, this tube over here is a 1:10 dilution. We had 1 part of our inoculum into 9 mils of sterile medium, which gave us 10 mils total. So this is a 1:10, and when we take 1 mil of this and add it to 9 more mils of sterile medium, that's a 1:10 of a 1:10. So what we would do is we would multiply these numbers together. So 1 times 1 is 1, and 1 times 10 is 100. So this is a 1 to 100 dilution. If we take 1 milliliter of this 1 to 100 dilution and we add it to a new test tube of sterile medium, we're basically going to have a 1 to 10 dilution of our 1 to 100 dilution. Okay, so if you multiply 10 times 100, what do you get? You get 1,000. So this is a 1 to 1,000. If we take 1 milliliter of that and add it to a new test tube containing 9 milliliters of solution, we're making a 1 to 10 dilution. I really should get a stylus. A 1 to 10 dilution of a 1 to 1,000 dilution. So this gives us a total of a 1 to 10,000. Remember, we're multiplying these two numbers together here. So we get 10,000. Then if we take 1 ml of that, add it to 9 ml of medium again, we're making a 1 to 10 dilution of our 1 to 10,000. And that gives us 1 to 100,000. So that's the final dilution. Next, we would take a sample of that and plate it onto a petri dish. So the next thing we would do is we would take a sample from each dilution and we would plate it on a petri dish. And there's a couple of ways that we can do this. The first way is the pour plate method, which is not that common in my experience. Basically what you do is you take 1 milliliter or about 100 microliters of broth from your dilution and you'd add it to a petri dish. Then you pour warm but molten nutrient agar on top of that and you'd spin the plate rather to mix the bacteria with the medium. Then you let it cool and incubate at the appropriate temperature for the bacterium's growth. And when you look at your colonies, you'd either have colonies growing on top of the agar or colonies growing in the agar. And this is okay, except some bacteria are heat sensitive, so mixing them with the melted agar might actually kill the bacteria. The second thing is, if we're looking at the characteristics of the bacterial colonies in order to identify them, the characteristics might be obscured for those bacteria that grow in the agar instead of on top of it. So I'm sure it's used somewhere, but in my experience, it's not that common. I'm more familiar with the spread plate method because this is one that I used to use all the time when I was working in labs. So here you take a sample from each dilution and you'd add it to the top of an agar plate. Then you'd use a sterile glass spreader to actually spread the bacterial specimen or sample across the agar plate. Then you incubate the plate. And if you've spread the bacteria out well enough, you'll get individual colonies where all of the colonies are on top of the plate, which makes it easier for study, and you don't have to worry about killing the bacteria because the agar was too hot. So let's, for the sake of this example, say that we used the spread plate method and we took a sample, a 1 ml sample, from each of our dilutions and added them to petri dishes. The next step would be to count the colonies that result from each bacterium. Typically you want to count less than 300 colonies, so more than about 25 to 30 and less than 250 to 300. The reason you don't want to count beyond 300 is the counts become inaccurate and it's really hard to distinguish whether all of the colonies are actually separate. You don't want to count less than 25 because then your numbers aren't very accurate because your sample size is so small. So we would look at our plates. We couldn't use the 1 to 10 dilution because you can't count individual colonies. There's way too much growth. Same for the 1 to 100 and 1 to 1000. When we get to the 1 to 10,000, however, there's quite a few colonies here, especially in comparison to the 1 to 100,000. So we probably wouldn't use this because that's less than 25. We definitely can't see all of our individual colonies for these other plates. So the 1 to 10,000 would be our best bet. Let's do a quick count to see how many colonies there are. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32. So there's 32 colonies here. So that's a pretty good number. So calculating the number of bacteria that were in our original culture is not really that hard. We would use this formula where we have CFUs per mil equals our dilution times the number of colonies counted over the amount of medium that we plated, the amount of our dilution that we plated. So remember, CFUs are individual cells or little clumps of connected cells. Okay, so we're going to figure out how many of those there are per mil. So in this example, our dilution was a 1 to 10,000. So our dilution factor is actually 10,000. And we're going to multiply that by the number of CFUs. And remember from the last slide, that was 32. We counted 32 CFUs on this plate. We're going to put that over how big our specimen was that we plated onto the plate. In this example, we would have used 1 mil, so this will be 1 mil down here. So 32 times 10,000 is 32,000, and then we get our CFUs per mil here. So we have 32,000 divided by 1, which is 32,000 CFUs per mil. That's not the only way that we can count cells, however. Another way that we could count cells in a culture is by using what's called the direct microscopic count. So instead of using CFUs, we're going to look at individual cells. For this type of count, you would need a special slide called a Petroff-Hauser counter, or you could use a hemocytometer. Basically, this is a special slide that has troughs etched into it, and then a grid also etched onto the surface. You would place a cover slip on top of the grid, and then you add your bacterial culture to the trough using a micropipette usually. The medium would run through the trough and under the cover slip, and then it spreads out over the surface of the, it spreads out under the cover slip. And the bacteria that are in the medium would be dispersed along the grid. So then, depending upon what lab you're in, how you count is going to be different. But generally, the idea is that you're going to take several boxes in the grid and you're going to count the bacteria in those boxes to get an average. So this, for example, might be one box, but some labs have different rules about if the bacterium is on one of these border lines, whether or not it can be counted, that type of thing. In any case, let's count these bacteria. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. 14 cells within this one box. To calculate how many bacteria in our culture that translates to, we would use this formula. So we have number of bacteria per mil is equal to the number of cells counted in our grid divided by the volume of the area that's counted. So remember, to calculate volume, you can measure the length times the width times the depth of that area under the cover slip, and usually that information is given to you in the packaging that comes with the slide. So for this example, the volume of the area that's counted is 8 times 10 to the minus 7 milliliters, and we counted 14 cells. So we would put 14 cells over 8 times 10 to the minus 7. If we do the division, we should get something like 17,500,000. And this is the number of bacteria per mil. Let's say it's not a bacterial culture that we're interested in counting. Maybe it's the amount of coliform contamination in a lake or a pond. For that, we would use something called membrane filtration. In membrane filtration, you have a special kind of membrane, and although you can't see it using your naked eye, if you were to put the membrane under a microscope, you would see that it has little holes or pores in it. Water can flow through these holes and pores, but cells, which are larger, have to sit on top of the membrane. They can't pass through those holes or pores. So we can essentially sieve the bacteria out of the water that we're testing. So in this example, you would take 100 milliliters of pond water, lake water, ocean water, what have you, and you would pull it through this filter using a vacuum. The bacteria would be retained on top, water would flow through the bottom. Then you take that filter, put it into a petri dish with growth medium, and incubate the cells. And you could look to see how many coliform, these are E. coli style bacteria, bacteria from the colon, that you find. In this particular example, I believe they're using liquid endomedium. And what it does is it causes E. coli bacteria and other coliforms to turn colors and take on a metallic sheen so that they're easy to distinguish from non-coliform bacteria. So then you'd count the bacteria, and then you'd put the number of colonies over the volume that you filtered initially, which would be 100 mils. So if we saw 32 coliforms, then it would be 32 bacteria or CFUs per 100 mils of water. The last method that we want to talk about is a turbidity measurement. You might remember from lab that turbidity refers to the cloudiness of a solution. To measure turbidity, we're going to use a special machine called a spectrophotometer. So that's SPECTROPHO TOMETER, spectrophotometer. And the layout for these devices is basically the same. On one side, you're going to have a lamp, and this lamp may give off light that's in the regular visible light spectrum, or it may give off light that's in the UV spectrum. In any case, we're going to have light that's going to shine on one side of our specimen. So our specimen is going to go in the middle here. On the opposite side, we're going to have a meter that's going to absorb any light that passes through our specimen here, and then it's going to register how much light must have been absorbed by our specimen on this dial. And we can look at the absorbance of light either as transmission. So transmission describes how much light actually passed through the specimen as a percentage, or we can look at absorbance, which is measured in optical density units. And the more turbid your specimen is, the greater its optical density and thus its absorbance. In this example that you're looking at on this figure, we have a clear medium. So all of the light is passing through the specimen, it's reaching the detector, and so we see that 100% transmission of light is occurring and that there's 0 optical density units being absorbed. Now we've got a turbid broth, and as you can see, as some of the light rays approach the specimen, they get refracted back. So you can see here This light is coming back. Some light gets deflected off in different directions, but only a small percentage of light actually passes through our specimen and reaches the detector over here. So we can see that the detector measures this as 20% transmission, meaning only 20% of the light is actually reaching the detector. Or if we're looking at absorbance, we have an optical density of 0.7. Now this is all well and good, but this doesn't give us how many bacteria there are in the specimen. To get that, you would need to do something called a standard curve experiment. In this experiment, you would grow a culture of bacteria and you would remove small amounts, small aliquots, every few hours. From the small aliquot, you would plate a portion of that material onto an agar plate and incubate, and then you would put a small aliquot in the spectrophotometer. After I don't know, 24 hours or so, you would observe the plate to see how many colonies there are on the plate. And then you could graph the number of bacteria versus the optical density. And actually, you might reverse these axes. But the point is, you would correlate the number of bacteria to the optical density or the percent transmission. And then you could use this curve to figure out how many bacteria there were in your initial specimen. So that's it for this video. We started out by describing 4 methods for measuring bacterial growth. We said that whatever method we're using for bacterial growth, they can be divided up into 2 groups. You have the direct methods that actually look at the individual cells or CFUs, and we measure those directly, like with the plate count, the direct microscopic count, and the membrane filtration that we talked about. And then you have the indirect methods like the turbidity measurements with the spectrophotometer. In the indirect methods, instead of measuring individual cells, we're measuring some characteristic of our bacterial growth. And then we're calculating from that the actual number of cells present. So historically, this part of this chapter has been a little bit more challenging for people to understand, so please don't hesitate to reach out to me with questions if you have them. I'm happy to talk about this with you during an office hour, or we could try it by text or email. In our next videos, we'll be tackling the differences between prokaryotes and eukaryotes when it comes to genetics. I'll see you in those videos.